Welcome back to a new video tutorial session from Biology Boost Up. Today in this video tutorial lecture we going to study about the recombinant DNA technology and its application. So now let's start. At first, the definition of recombinant DNA technology. The deliberate modification of an organism's genetic informations by directly changing its nucleic acid genome is called genetic engineering. The genetic engineering is accomplished by a collection of methods known as recombinant DNA technology. First, the DNA responsible for a particular phenotype is identified and isolated. Once purified, the genes or gene are fused with other pieces of DNA to form recombinant DNA molecule. The term recombinant DNA usually refers to the creation of new combination of DNA molecules that are not found together naturally. Now, the basic steps in recombinant DNA technologies. The recombinant DNA technology is powerful tool for the isolation of potentially unlimited quantities of a gene. The basic procedure involves the following steps. At first, the purification of DNA from cells or tissues or construction of cDNA or complementary DNA from total RNA or poly a messenger RNA or mRNA using the enzyme that is called reverse transcriptase. Now the second steps is production of DNA fragments for genomic DNA only using the enzyme called restriction endonuclease that recognize and cut the DNA molecule at specific nucleotide sequence. Next, the joining of DNA fragments or cDNA using the enzyme that is called ligase to other DNA molecule that serve as vector or carrier molecule having capacity of independent replication. For joining genomic DNA fragments, cutting the target DNA and the vector will be done by the same restriction enzyme. The vector with an inserted DNA segment is called a recombinant DNA molecule. Now the next step is transformation of the recombinant DNA molecule carrying an inserted DNA fragment into a host cell in which the chosen molecule can undergo replication independently of the host cell chromosome and produce dozen of identical cells that is called the clone. As host cell replicate, the recombinant DNA molecule within them are passed on to all their progeny cells, creating a population of host cells, each of which carries copies of a cloned DNA sequence. Now the last step is isolation of recombinant DNA clones by harvesting the expanded cell cultures. Subsequent purification and analysis of that target DNA. Potentially, the cloned DNA can be transcribed, its mRNA translated, and the gene product isolated and used for research or sold commercially. Now, the application of recombinant DNA technology. First point, DNA technology is also used to detect the presence of HIV in a person. Next, application of recombinant DNA technology in agriculture. For example, manufacture of BT cotton to protect the plant against bollworms. Third point, application of medicines. Insulin production by DNA recombinant technology is a classic example. Next, gene therapy. It is used as an attempt to correct the gene defects which gives rise to hereditary diseases. Next, clinical diagnosis. ELISA is an example where the application of recombinant DNA technology is possible.